My voice is. Hello. Hello. Okay. Is it okay now? Yeah. What you have discussed yesterday is bacteria general characters, bacteria structure, difference between the archaea and bacteria, archaebacteria and bacteria, and difference between prokaryote and eukaryotes, difference between the prokaryotes and eukaryotes, and also discuss the what we have discussed and types of the bacteria based upon the flagella. Flagella types, difference between prokaryote and eukaryotes, flagella types, and structure based upon the structure, various types of the bacteria we have studied, and bacteria cell structure also we have studied. Bacteria cell structure, what are those parts are there in the bacterial cell structure that we have discussed? Capsule, cell wall, plasma membrane, cytoplasm, reserve food material, nucleoid, plasmid. This all we have discussed. You see here nucleoid, how it is, how the nucleoid is, it is a naked. There is no nuclear membrane, no chromatid structure. This DNA, single long circular strand DNA is there, not the double strand DNA. Single circular DNA is there. That we call it as bacterial chromosome, also called as genophore. What I told you, also called it as genophore. Bacterial chromosome or genophore. Okay. There is no histone protein, there is no chromatid structure. That all we have discussed. Flagellas also we have discussed. Monotrichous and amphitrichous, lopotrichous, peritrichous. Okay. Atrichous, peritrichous. We have studied. Atrichous means there is no flagella. Okay. With the flagella, it can move. Usually, flagellas are present in the gram-negative bacteria. Okay. Amphitrichous means the both end flagella is present. That is amphitrichous. We have studied. And peritrichous means the both whole surrounding of the cell wall flagella is present. That is peritrichous. Okay. These are the types we have studied. And along with the flagella, we also studied the villi. We also studied the villi. You know the difference between the flagella of the bacteria and other plants. Bacterial flagella is made up with fimbrian protein. Fimbrian protein 9+2 arrangement. Where you see the villi. These villi are two types, normal villi and sexual villi. These villi are straight and short, straight tubular structure. This will be useful for these are useful for the conjugation. The conjugation. That means for the fusion of plus and minus chain bacteria, F plus and F minus. For the sexual reproduction. In the sexual reproduction, one of the most important reproduction in the bacteria, that is conjugation. In that conjugation, the villi are more important. These are useful in the sex. That's why sexual reproduction. That's why we calling that sexual villi. Okay. And we also can see the structure of capsule. I already told you, extra layer, outer layer above the cell wall, that is made up of it polysaccharides and polypeptides. In the cytoplasm, here it is the middle part, is the cytoplasm, the whole structure of the cytoplasm that we have discussed already. Where it is? Okay, that all we have discussed. Then we will go. You see how it is. In the cytoplasm, 80% of the water is there, 20% of salt and proteins are present. DNA is in circular, haploid. You see, only single circular DNA is present. There is no histone protein. And at the middle, what are there? You see here, along with the nucleoid, bacterial chromosome, plasmids also present. The plasmid, extra DNA, extra chromosomal DNA, extra nuclear DNA, extra chromosomal DNA, double helical, circular, self-replicated, extra chromosomal DNA is called as plasmid. I told you, plasmid first identified, given the name by Nirenberg. I already told you. Okay, and then what you have discussed, the plasmid types also we have discussed. No cell membrane organelles are present. Cell membrane organelles are absent. Mitochondria, chloroplast, Golgi bodies, endoplasmic reticulum, these all are absent. Only the ribosomes are present. Okay. What are the other cytoplasmic components which are there in the bacteria cell? That are mesosomes. I told you the mesosome. The plasma membrane of the bacteria fold inside. Plasma membrane of the bacteria is fold inside, and it form the special structure, the lobe structure that we calling it as what it is? Mesosome. That special structure is called as mesosome. Okay, here you can see this mesosome is a folded plasma membrane, folded one is the mesosome. This will be acting as a mitochondria, respiratory. It having the respiratory enzyme, that's why it will be acting as a mitochondria. These are most prominent in the gram-positive bacteria. You see, these are the intracytoplasmic inclusion. This goes inside the cytoplasm. It enter into the cytoplasm. Whatever the plasma membrane, that will be the bonding proteins of the mitochondria. It also will be useful for the fission, division of the one bacteria in cell into two. You can see the cell membrane, how it is between the gram-positive and gram-negative bacteria also we have discussed. Cell membrane. Cell membrane, here you can see the more phospholipids are present in the gram-negative or peptidoglycan, more peptidoglycans are present in the gram-positive. That all we discussed, difference between the gram-positive and gram-negative bacteria. You see, peptidoglycan polymer layer is present. That is made up of it NAG and NAM, N-acetyl glucosamine, N-acetyl muramic acid. NAG and NAM, these two are present. And you see the lipopolysaccharide structure how it is. And then you can see the difference between the gram-positive and gram-negative bacteria that also I told you. Here in the gram-negative, lipopolysaccharide membrane is more. In between the lipopolysaccharide, peptidoglycans are present. Whereas in the peptidoglycans are more, along with that, lipotoic acid, mevalonic acid are present in the gram-positive bacteria that are not there in the gram-negative bacteria. But more lipoprotein membrane are there in the gram-positive bacteria. Okay. And you can see the plasmid, how it is. Plasmid is a bicircular extra chromosomal DNA, self-replicated extra chromosomal DNA. This is the plasmid. And also we studied the types of the plasmid, conjugated plasmids and non-conjugated plasmids. You see, it are useful in the conjugation and exchange of the genetic material in between the two that are conjugated plasmid, that we call it as F1 F plasmid. F plasmids. If the plasmid having bacteria, we calling it as F plus. If there is no plasmid, that we are calling it as F minus. In the conjugation, the non-conjugated plasmids, these are various types are there. That are 5 types. Basically, plasmids are 5 types are there. What are those? F plasmid that I told you. Plasmid which are exchanging genetic material from one to other in the conjugation. R plasmid, these are shown the resistance to the antibiotic. Resistance. It's shown the resistance to the bacteria that we call it as R plasmid. You see, and then coliplasmids, these are there in colibacteria. Okay, these are colibacteria which release the toxin proteins that cause for the death of the bacteria. Okay, that coliplasmids which cause for the death of other bacteria, and degradative bacteria and degenerative bacteria. These are there in the Pseudomonas foetida. You know Pseudomonas foetida? We use Pseudomonas foetida in the degradation of the plastics in the ocean, or even for the debug of the oil. We use the Pseudomonas foetida. For the cleaning of ocean oil and plastic material, we use the Pseudomonas foetida. And some of the here and there, some of the endospores also present. These are resisting spores. This can be regenerate their own thallus, their own bacteria after whenever the harmful situation there, then it will be remain, the endospores will be remain, but the remaining cells will be, even remaining cells die. These endosporic cells will be remain, that will be regenerate their own thallus. That will be regenerate their own thallus, that are calling it as, that we are calling it as endospores. Endospores. Okay. Then where we are, slide share is not there. Okay, again I have to share. Yeah, here you can see. Yeah, now you can see. Okay. Here we already studied the difference between the gram positive and gram negative bacteria that we have studied. Okay, is it visible? Or otherwise I will make it 100. Okay. Then you can see here easily difference between the gram positive and gram negative bacteria that we already studied. And now we will go for this all we have studied. We will go Further, next flagella also we have studied. And next is nutrition. Nutrition in bacteria. There is a two type of the nutrition, that is autotrophic and heterotrophic. Autotrophic nutrition and heterotrophic type of the nutrition. Autotrophic. Autotrophic means they can synthesize their own food material. They can synthesize their own food material either by selection to control it. Selection. Okay. The autotrophs and heterotrophs. These are the two types. You can see autotrophs means they can produce their own food material. They can synthesize their own food material. Heterotrophs means it depend upon others. That all we have studied in the fungi. In other chapters also we have studied. Autotroph, again two types. Photoautotrophs and chemoautotrophs. Photoautotrophs and chemoautotrophs. That means if it is depending upon chemicals for their synthesis of food material, that we call as chemoautotropic. If it is depending upon the light, as like other plants, photo means not photo, taking photo. Photo means light. Light. Photosynthesis. You know the photosynthesis, synthesis of the food material under the light. That is photosynthesis, basic life-giving reaction. No, like that, here also photosynthetic bacteria are also present, that we call as photoautotroph. Photoautotroph. Okay. Photoautotrophs and chemoautotrophs. Photoautotrophs and chemoautotrophs. You can see here, it is not moving. Okay. Autotrophic bacteria, what are those? What are those autotrophic bacteria? I told you now just. Chemoautotrophs and photoautotrophs. Okay. Chemoautotrophs and photoautotrophs. Here you can see they are synthesizing their own food material. That's why we are calling it as autotroph. They are synthesizing their own food material. That's why we are calling it as autotroph. But how, where it is depending? Depending upon, we divide it into two types. Depending upon the source, we divide it into two types. That is photosynthetic and chemosynthetic. That means energy utilizing either from the light or from the chemical. Okay. The photosynthetic bacteria, you can see there is a various type of the photosynthetic bacteria are there. You see, in the photosynthesis, what happened actually? Photosynthesis, what happened? It absorb the carbon dioxide and water, it release the oxygen. Okay. The oxygen. And also synthesize the carbohydrates. Okay. Here also, the bacteria which are absorbing the solar energy in presence of photosynthetic pigment. I told you the photosynthetic pigments, how many types of the chlorophyll pigment? 9 types. Chlorophyll A, B, C, D, E. Chlorophyll A, B, C, D, E. Along with that, in the bacteria, what are there? I told you bacterial chlorophyll A and bacterial chlorophyll B. Bacterial chlorophyll A and bacterial chlorophyll B, and also Chlorobium, chlorophyll 650, and Chlorobium, chlorophyll 660. These four, bacterial chlorophyll A, B, and Chlorobium, chlorophyll 650 and 660, these four are present in the bacteria. These four are present in photosynthetic bacteria, not in all the bacteria. These four are present in the photosynthetic bacteria. There are bacterial chlorophyll A and bacterial chlorophyll B. Okay, Chlorobium, chlorophyll 650 and Chlorobium, chlorophyll 660. With that chlorophyll pigment only, they could able to synthesize the food material. They could get their own food material. They could get energy from the light. Okay, but one difference between the bacteria and other plants. What is the difference? Do you know? Based upon that one, experiment also is there. Hill reaction. You know the Hill reaction in the photosynthesis? You might have studied in the fourth paper. Hill reaction. There is an experiment. You know, Hill-Bendall reaction. To identify the oxygen, where from the oxygen is releasing. Either from carbon dioxide or from water. Either from carbon dioxide or water. To know there is a Hill reaction is there. With that, we could able to know oxygen is evaluating from the H2O, water, H2O, water, not from the CO2. Okay, not from the CO2. It is releasing from H2O. The H2O oxygen is separating, hydrogen molecule is separating. That hydrogen molecule is utilizing in the energy, ATP synthesis. Okay, but not from the carbon dioxide. It is releasing from the H2O. Okay, here you can see mostly these are all sulfur bacteria. Sulfur bacteria, it's not utilizing the H2O. Instead of H2O, these are utilizing H2S, hydrogen sulfide. You know the hydrogen sulfide, it having fall egg smell. Kullina godi gudla vasana smell. Fall egg smell. You know that sulfur bacteria which are participating in the photosynthesis, but it releasing sulfur, not the oxygen. Why? Here instead of H2O, H2S is participating. That's why instead of oxygen, sulfur is releasing. Based upon this reaction only, we could able to know oxygen is evaluating from the water molecule, not from the carbon dioxide. By doing the experiment on these bacteria. Okay, you see, there is a three types: green sulfur bacteria, purple sulfur bacteria, and non-sulfur bacteria. Competitive exams, so many questions are asking which are the sulfur bacteria, which are the green sulfur bacteria, which are the photosynthetic bacteria. That is Chlorobium, Chromaceum, Rhodopseudomonas. You see, green sulfur bacteria is what it is? Chlorobium. What is it? The Chlorobium. Chlorobium is the green sulfur bacteria, and Chromaceum is purple sulfur bacteria. That means it is in purple. Photosynthetic pigment is in purple in color. That's why purple sulfur bacteria. It is in green in color. That's why green sulfur bacteria. Why we are using sulfur bacteria? It is releasing the sulfur. It is using the H2S instead of H2O. And non-sulfur bacteria. Okay. Here it is not using the sulfur as a byproduct. Instead of sulfur, it is as using the H2O, that is Rhodopseudomonas. Rhodopseudomonas bacteria. Okay. And then you can see The chemosynthetic bacteria. What are those? Chemosynthetic bacteria. That chemosynthetic bacteria, can I zoom it? Otherwise, it is not clear to you. Okay. Now it is very clear. Okay. Chemosynthetic bacteria. That means it depending upon chemicals for their energy as an energy source. As an energy source, they are depending upon the chemicals. Based upon the chemicals, there is a various type of the chemosynthetic bacteria are there. Based upon the dependency of the chemicals, whether it is using the nitrates or phosphorus, iron, hydrogen, ammonia, based upon the which chemical, in this reaction, what happens? Chemical bonds will be broken. By breaking the chemical bonds, by breaking these chemical bonds, it releases the energy. It releases the energy. Chemical energy will be released. That released energy will be utilized by the bacteria. That's why these are we calling it as chemosynthetic bacteria. First, you can see the sulfur bacteria. Thiobacillus, bigiata. You know the largest bacteria is bigiata. Thiobacillus and bigiata. These are utilizing the sulfur byproducts and releasing the sulfur and energy. It is depending upon the sulfur compounds. That's why we call it as sulfur bacteria. You know, just we have studied green sulfur bacteria and purple sulfur bacteria. Based upon this reaction only, we could able to know oxygen is releasing from H2O, not from CO2. This reaction based upon that is Hill reaction. In the physiology we have studied. And iron bacteria, which is depending upon iron product, iron components, it oxidizes. It do the oxidation reaction. By oxidation reaction, it is releasing, it absorbing the oxygen and releasing the carbon dioxide and energy. Carbon dioxide and energy. This iron bacteria, sulfur bacteria, iron bacteria. Iron bacteria we also call it ferrous. And nitrifying bacteria. You know the nitrifying bacteria are two types. Nitrosomonas and Nitrobacter. Nitrosomonas and Nitrobacter. You can say here Nitrosomonas, it converts atmospheric nitrogen into NO2. Atmospheric nitrogen into NO2. N2 gives rise to, in presence of oxygen, it is converting into NO2 nitrates. N2 to NO2. That is that reaction by Nitrosomonas. And by the Nitrobacter, NO2 in presence of oxygen converting into nitrites, NO3. NO3, NO2, N2 to NO2, NO2 to NO3. Nitrogen to nitrate, nitrate to nitrites. There we are releasing the energy. Because of these nitrifying bacteria only, we could able to get the protein. We even 78% of the nitrogen is there in the atmosphere. We can't utilize that 78% of the nitrogen, N2. That will be utilized only by the bacteria, rhizobium, and some fungi, some blue-green algae. Bacteria only utilize this atmospheric nitrogen. It converts into nitrates, nitrites, and ammonia. That ammonia and nitrates will be absorbed by the plants and it synthesizes the amino acids and proteins. That amino acids and proteins we will get from the plants, directly or indirectly. due to these nitrifying bacteria only. Okay, sulfur bacteria, iron bacteria, hydrogen bacteria. If it is depending upon hydrogen, hydrogen bacteria. Hydrogen in presence of oxygen, it form water molecules and energy is released. Hydrogen bacteria, Pseudomonas, Hydrogeomonas, Bacillus, Spectrotropa. These are we use for the cleaning reactions, cleaning purposes. That all are there. These all are the, what are those? These all are autotrophs. These all are autotrophs. And again, we will discuss the heterotrophs. Next is heterotrophs. Autotrophs, autotrophs two types, photoautotrophs and chemoautotrophs. Photoautotrophs three, sulfur bacteria, non-sulfur bacteria. Chemoautotrophs based upon the chemicals depending upon iron bacteria, sulfur bacteria, hydrogen bacteria, and nitrogen bacteria. There is a difference between photosynthesis and chemosynthesis. Here you can see energy is very less in the chemosynthesis than compared to the photosynthesis. Here energy is gained by splitting the chemical compound, releasing the chemical bond. Here it is getting energy from the planet sun source. Here no light is involved, but here totally sunlight depending upon the sunlight. Here reactions, that is exothermic, energy releasing reaction. Here it is a reaction as endothermic, energy fixed result inside. Energy required process obtained by oxidation of some organic, inorganic, organic substances in the environment. And but here in the solar energy, for energy depend upon solar energy. And no pigments are present in the chemosynthetic bacteria, but bacteria chlorophyll and chlorobium chlorophylls are present in the photosynthetic bacteria. You know, the purple sulfur and green sulfur bacteria, Pseudomonas, Theobacillus, that are participate in the chemosynthetic for the degradation of sulfur bonds and it convert into sulfurs, sulfur. You know, foul egg smell to good smell, sulfur, gandham smell. You see, and here in the photosynthetic bacteria, what happen here? It is observing the energy from the sunlight. Okay, and then next is the heterotrophic bacteria. That means it's depending upon others. Heterotrophs again, two types of the, saprophytic and parasitic. Saprophytic bacteria means which grown on, saprophytic bacteria means which grown on dead
and decaying matter.
Organic matter. Which grown on dead and decaying organic matter, and it digests and it do the cleavage. That's why we are also called as scavengers. It cleaning the environment. It break down the complex organic matter, proteins, lipids into simple minerals. Mineralized humidification will take place by these saprophytic fungi and bacteria. Humidification, humus formation in the soil. So by breaking the proteins and lipids, it get converted into minerals, nitrogen, phosphorus, like that, manganese, magnesium, nitrogen, phosphorus, that will be separated and it will be added to the soil. And then the parasitic bacteria means which grown on leaf. Live, that means either living plants or an animal, living organism. Living organism, it depends upon living organism. It may cause the disease harmful to the host. sometimes, the symbiotic bacteria is also there. Now, E. coli bacteria and rhizobium bacteria. E. coli bacteria, E. coli bacteria which is symbiont with our intestine, which is there in our intestine, which will make our food to alkaline so that it's easy to digest. To maintain the pH level of the food, it will be useful. Otherwise, we can't digest our intake food material. There is a need of fermentation of the food in intestine. That fermentation will take place by E. coli bacteria, symbiotic bacteria. Other symbiotic bacteria, rhizobium bacteria, which are present, which are associated with the root nodules. Root nodules, especially in the pulses, Leguminaceae family. Leguminaceae family roots are having associated with the rhizobium bacteria. Rhizobium bacteria. This rhizobium bacteria absorbs atmospheric nitrogen and it converts into ammonia, nitrates and ammonia, so that this ammonia will be utilized for the production of proteins, amino acids and proteins in the plants. Amino acids and proteins in the plants, so that this will be useful in the synthesis of proteins in the plants. That are two are symbiotic type of the nutrition. What are those two symbiotic type of the nutrition? E. coli in human intestine, rhizobium associated with the pulses. That's why the pulses having the highest, richest proteins, rich protein, because of this rhizobium bacteria, which are absorbing the atmospheric nitrogen and get converting into ammonia. That ammonia is absorbing by the plant and it making to amino acids. This amino acid participating in peptide bond so that it is forming the proteins. In the protein synthesis, rhizobium bacteria are being utilized. That's why more proteins are synthesized in the pulses than the other plants. Soya bean having the highest, richest quantity of the proteins. Soya bean having the richest quantity of the proteins. Why? That's the reason. Okay, and then the parasitic. There's so many diseases: cholera, dysentery, cholera, typhoid, pneumonia. These all are the diseases caused by the bacteria to human beings. Typhoid, cholera, dysentery, diphtheria, tetanus. These are so many diseases which are causing by the bacteria. Bacterial diseases. There's so many plant diseases also. Plants also, crown gall diseases, necrotic diseases. So many diseases causing by bacteria to the plants. That all are parasitic. That all are parasitic bacteria. Okay. Photosynthetic bacteria, autotrophic bacteria, heterotrophic bacteria. Autotrophic bacteria again divided into chemosynthetic bacteria and photosynthetic bacteria. Heterotrophic bacteria divided into three. What are those? Saprophytic bacteria, parasitic bacteria, and symbiotic bacteria. What are those? Symbiotic bacteria. Okay. These are the three types of heterotroph. These are the nutrition in the bacteria. Okay. A question they asked. Write about the types of the nutrition in the bacteria. And next is reproduction, also very, very important. In the bacteria reproduction. Bacteria reproduction is very important. You see, asexual reproduction and sexual reproduction. Asexual reproduction and sexual reproduction is there. Asexual reproduction by especially vegetative and sporulation. Vegetative reproduction by fission, that means division into two. Division into two, and one bacteria division into two bacteria. And budding, you know the budding in the East, we have studied the budding, forming the birds, and cysts, forming the cyst upon the earth, and segmentation, that is branches formation. These all are the vegetative type of the reproductions which are there in the bacteria. And the sporulation, conidia formation, ioiospores, arthrospores, small minute spores formation, and endospores, spores form inside the bacteria, endospores. And the next is sexual reproduction by three ways: the conjugation, transduction, and transformation. Here, the sexual reproduction, it is not the true sexual reproduction in the bacteria, but genetic material is exchanging from one to other. That's why we're calling it as sexual reproduction. But there is no male and female bacteria. There is no sexual variation. The organs in the bacteria. They are participating as opposite stain only. One is a plus stain, one is a minus stain. Here, genetic material is exchanging, but there is no fusion, there is no syngamy type, there is no sexual organs in the bacteria, but it pseudo-sexual reproduction. That means genetic material is exchanging. That's why we calling it as sexual reproduction, but it is not true sexual reproduction. You remember, it is not the true sexual reproduction. Okay? You see, there is a various method of conjugation. A plus, F minus, and HFR, A plus. Thus, we will discuss. Here, we will discuss various types of the sexual reproductions. Reproductions type, vegetative, asexual, and sexual reproduction that we will discuss here. You can see. Now I can't now. To close it. Okay. Then you can see here reproduction, asexual reproduction. Asexual reproduction, two ways: vegetative reproduction by spore formation. I already told you binary fission. The normal type of the asexual reproduction in the bacteria is binary fission. To multiply within the short span of the time, the bacteria will take the binary fission. So in the so many competitive exams, suppose if bacteria, it takes for the multiplication, it takes 30 seconds. After 2 minutes, how many number of the bacteria are present in the sample? Are you there in the class? Can anybody say? Multiplication will taking place by the fission, binary fission. One bacteria is forming two bacteria. Within the 18 seconds, one binary fission will take place 18 seconds. That means in the 1 minute, how many? How many number of the bacteria will form in the 1 minute? Respond. Are you not there? If suppose one bacteria, 18
8
seconds.
bacteria. 8 bacteria.
8 bacteria, yes. That's good. If suppose another very easy and very toughest question in the competitive exam. If suppose the bacteria culture is off in the, suppose in the 58 minutes or in the 59 minutes. 59 minutes, bacteria culture is off, and then up, but and after 1 hour, how much quantity of the bacteria is present in the culture? In the 59 minutes, it is off. The 60 minutes is 1 hour, 60 seconds is the 1 minute. Okay. In the 59 minutes, bacteria culture is off in the sample. How much quantity of the bacteria there in the culture after 60 minutes?
It is full, sir.
It is?
Full.
Yeah, I already drawn here. It is full. Here it is half. Why? It is taking only 1 minute for the multiplication. Yes or not? It is taking within the 1 minute is multiplied. So before 1 minute it is half. After 1 minute is multiply means 50 plus 50, 100. Full fledged. It is competitive exam. This exam asking so many competitive exams. If the bacteria is multiplying in 1 minute, in the 59 minutes or at 58 minutes, in the 58 minutes, it is only 25% water. At the time of 60 minutes, how much quantity of the bacteria is present in the sample? In the 58 minutes, 25. 59 minutes, 50. 60 minutes, 100. Okay, have you understood? There is a question asking so many times in so many competitive exams. A mental ability question. Okay. You see, the next is budding. That's why, for what purpose I am saying here is, here it is taking multiplication very rapidly. If the one bacteria is there, if you neglect one bacteria, within the minute, it is multiplies. Within the hour, it multiplies how many times? Not the 60 times, more than 300 times. That much of the multiplication is taking place. Okay. And then here you can see the next is budding. You know the budding. That means formation of the buds. Formation of the birds, budding. Okay. And budding means I already told you in the yeast how it is taking place. At the apical part, it's swollen. In the swollen part, whatever the material, genetic material, cytoplasm will go into that is forming the bubble-like structure. If even it split from the mother cell, it will become the new bacteria. That are there in Hypomonas, Podomicrobium, or Rhodopseudomonas. There's so many bacteria. You see, even the bacteria is forming the bud. To this, the cytoplasm and genetic material will enter. Like that, it is forming the body. Even it split from the mother, it will become the new bacteria. I insist. Sometimes, some of the bacteria cytoplasm is forming rigid, thick-coated cell wall and rounded with entire protoplasm to escape from the adverse conditions, drought condition, adverse condition, abnormal condition, dry condition. It making the cysts rigid hard coated cell wall, so that it will be prevent from the damage. It is similar to endospores, but endospores, it is whatever the cytoplasm genetic material, it is forming the spore inside. But all the bacteria is forming as a cyst here outside. Whatever the outer cell wall is forming thick coating. Endospores and cysts and segmentation. That means segmentation means separation. One body is forming into two small bodies. Gonidias or conidias like structures. Whenever the favorable condition that splits and each one will become the new bacteria. Okay. This is the vegetative asexual type of the reproduction. And you can see the binary fission, how it is taking place. The binary fission, one bacteria is dividing into two by the binary fission. And you can see the budding, how it is, budding is taking place. And you can see the cysts, how the thick coating, bacteria cell is thick coated, cyst formation is taking place inside. And you see the segmentation, division taking part, septors formation, segmentation. Each one will become the new bacteria. And you can see the sporulation. Sporulation also is taking place, is forming the conidia, iodia type. That these are especially in the actinomyces bacteria, filamentous bacteria, fungi-like bacteria, which are present in the fungi-like bacteria, is called as actinomyces bacteria, actinobacteria. Conidias, iodias are those spores. Small, minute spores are called as iodiospores. And you also can see the sporangiospores. Sporangiospores, especially these all are there in the filamentous bacteria, actinomyces bacteria, not in normal bacteria. Actinobacteria, actinomyces. Actinomyces means these are gram-positive bacteria which are look like the fungal filamental-like structure, fungi filamental-like structure, but is a bacteria, prokaryotes. That's why we call it actinobacteria, actinomyces. That are all shown the various type of the sporulation. What are those type of the sporulation? Conidia, idiospores, sporangiospores, and you can see the endospore, formation of the spore inside, that is endospore. Formation of the spores inside, that is called as endospore. What happened? It is not moving. Okay, you see here the endospores, how the endospore formation is taking place. Inside, it will be prevented from the damage, dry, drought condition, abnormal condition, and whenever it's wet, then it is forming the new bacterial cell. That is the endospore. And then the next is sexual reproduction. Here there is no sexual organ, there is no fusion, syngamy is like reproduction, plasmogamy, karyogamy like that not happening. But here genetic material is exchanging between the two organisms. That's why we call it as sexual reproduction. These are parasexuals, not the perfect sexual reproduction. Equal to sexual reproduction. Why? The genetic material is exchanging. That's why we call it as sexual reproduction. But not sexual organs. No sexual organ. You can see the three types, that is conjugation, transformation, transduction. Conjugation, transformation, and transduction. The majorly happening type of the sexual reproduction is conjugation. In the genetic material exchange, the conjugation is one of the criteria that with the sexual villi, sexual villi we have studied. The villi and sexual villi. The conjugation first observed, identified in E. coli, Escherichia coli by Ledenberg and Totem. Very, very important in the so many competitive exam. Asked the question. Conjugation observed, first discovered in E. coli, pneumonia, Streptomonas, like that they ask the question. In which bacteria they are identified, they discovered the conjugation? E. coli. Conjugation first discovered by whom? Ledenberg and Tatum. I told you the Ledenberg given the name plasmid. Plasmid, extra chromosomal DNA. Self-replicated extra chromosomal DNA is called a plasmid. Plasmid types also we have studied. F plasmid, R plasmid, coli-plasmid, degenerative plasmids. Various type of the plasmids we have studied. The plasmids first identified, given the name by Ledenberg. By observing this reaction only, by observing the conjugation reaction, he could able to know there is an extra chromosomal genetic material is there, not the chromosomal genetic material, other than the chromosomal genetic material also exchanging from one organism to other. That is conjugation. Conjugation, you know, even in the spirogyre also you have studied the conjugation. Forming the tube-like structure in between the two opposite states. Here you can see the what happened in the conjugation. You see two opposite stain, that means plus F plus and F minus. F plus stain and F minus stain. F plus mean, can anybody say F plus mean?
Plasmid is present.
Yeah. In which organism plasmid is present, that one we calling it as F plus. If plasmid is absent, that we call as F minus. F plus and F minus. If plasmid is absent, that we calling it as F minus. The plasmid is present, that is F plus. Here, genetic exchange is taking place only in the plasmid gene, not the actual genetic material is exchanging from one to other. Only the plasmid genes are exchanging from one to other. F plus plasmid and F minus plasmid, there is a sexual phyllide. The sexual phyllide, tube-like, shortened filament, that we calling a sexual phyllide. With the sexual phyllide, the two opposite strain, F plus F minus will be attracted. The sex phyllide will attach in between the two, F plus and F minus. F plus and F minus cells. These are rough. Otherwise, it is fully in between the F plus and F minus. You can see here what happening here. What is happening?
Transfer of plasmid is
whatever the plasmid that is fitted, double helical plasmid that is fitted double helical plasmid, you see here it is going to F minus, going to F minus through the conjugation bridge from F plus to F minus, so that the whatever the plasmid that is whatever the genetic material that also present in the F minus, so that it also will become F plus. Now it is not the F minus. Why? It also having the plasmid. Here what happened? Genetic material is exchanged in between the two organisms. That's why we call it as sexual reproduction, but it's not the perfect sexual reproduction. Okay? Okay, have you understood?
Yes, sir.
You see, and this conjugation is also taking place not only F plus F minus, even it is conjugated bacteria along with normal bacteria also. Whenever the conjugated bacteria, it having the extra chromosomal gene, which are replicated gene, that we calling also already told you, episome. That HFR plus F minus. What happening in the HFR plus F minus? What happening? Here this episome is mixing with the genetic material, actual genetic material. Actual genetic material, you see this one. Actual genetic material is mixing with the plasmid genetic material. So that we are calling it as HFR, high frequency recombinant DNA. Why? What it is happen? By mixing of the episome, that means plasmid genetic material with the nuclei. You know, this will be happen in the lambda bacteriophages, especially viruses, lambda bacteriophages. The T2 bacteriophages, lambda bacteriophages you have studied, the viruses. That also we studied in the next class, the virus subject. So here the genetic material also, whatever the mixed with the plasmid, that is entering into the F minus. So that what happened? 2 will become HFR. These are the two type of the conjugation between the F plus and F minus and HFR plus F minus. Okay, HFR minus F is exchange of the normal genetic material also along with the plasmid. Normal genetic material also being exchanged. That is HFR, high frequency. That means fertile male with female. Fertile male. That means HFR with F minus. This will be obtained in the lambda phase, bacterial phase, especially in the transduction phases. It are mixed. HFR means mixed with the normal genetic material. It means that completely deployed. And the next is transformation. Transformation. Transformation means transformation. Transformation. You know, the transformation means transformation, transduction. Transformation means transformation. Here, the transforming the genetic material from one to other. Transforming means exchanging, transferring, moving, moving of the genetic material from one to other. You know, this mechanism is first discovered by Frederick Griffith. Who is that? Frederick Griffith. Frederick Griffith, it is especially in pneumonia bacteria, pneumonia causing bacteria. Clostridium pneumoniae, pneumonia causing bacteria. We observe the pneumonia, you know, the pneumonia disease infect from other environment also, from outside, whatever it is, the living organism which have pneumonia bacteria, if it is enter inside to our body, we cause the pneumonia disease. Yes or not? Here, without any carriers, the genetic material is exchanging from one to other. He has done experiments on the mouse. He has done experiments on the mouse. On the mouse, what he has done, he injected sterile pneumonia bacteria. That means disinfected pneumonia bacteria into the mouse, and mouse is very live. There is no effect to the mouse. It is very active. And then he also injected the pneumonia causing active genetic material into the mouse. Active genetic material into the mouse. Then what happened? That mouse has died by affecting with the pneumonia. And then later, next, what he has done, he has injected the attenuated, sterilized genetic material along with the non-sterile, the mixed. That time also, what happened? The mouse are died. Why? Genetic material is exchanging from the living, even from the active genetic material will be attenuated, again reactivated, and it caused the disease. It caused for the death of mouse by pneumonia. Pneumonia causing bacteria. He has done experiment on pneumonia causing bacteria. Who is that? Frederick Griffith. Transformation experiment done by whom? Frederick Griffith.
Griffith.
On which bacteria?
Pneumonia bacteria.
Pneumonia causing bacteria. Okay. With this experiment, he could able to know how the disease are affecting the living beings, how the disease is causing, how the disease is spreading. from the environment or from the water from outside the living organism to inside. How it is happening? He has done the experiment. You see how it is happened. He has done that experiment. You know, for that experiment, he got the Nobel Prize also. And the next is transduction. That means foreign genetic material, and whatever it is, the foreign genetic material, other genetic material transforming into the living organism. You see here, transduction is causing by carriers. Special carriers are there. In presence of the carriers only, it will be moving from one to other. The transduction first discovered by Ginder and Ledenberg. Ginder and Ledenberg on Salmonella typhimurium, typhoid causing this bacteria. Salmonella typhi, typhimurium, typhoid bacteria. What he has done, you know, here in this experiment, you might have studied the replication in the viruses. Replication of the virus, that is same reaction in the transduction. You see, virus here, the genetic material exchanging through the viruses, through the carriers. Here, what are the carriers? You see
Bacteriophage.
T2 bacteriophage. This is penetrating whatever the genetic material it is, whenever it attached to this, it is entering into the genetic material, enter into the bacteria. This is multiplying and is attached with the bacterial genetic material, and it kill the total, disintegrate the bacterial genetic material, and totally it multiply the viral genetic material. So that after few minutes, bacterial genetic material will be disintegrated, totally degraded, only viral genetic material will be remain. And after some time, the bacterial cell wall is splits and all the viruses, number of the viruses will come out. You know the lysis and lysogenic cycle, two cycles. If the bacteria is killed, that is lysis cycle. If the bacteria genome also is there along with the viral genome, that is lysogenic cycle. In the both, the genetic material is exchanging, transforming by the other carrier. The other genetic material is carry forwarding in the bacteria, that we calling it as transduction. Here, exchange of the genetic material by carriers is called as transduction. Exchange of genetic material by the other carriers is called as transduction. If exchange of genetic material, transformation of the genetic material from the environment, that is transformation. Conjugation means by fusing of the two opposite plus and minus chain, that is conjugation. By these three ways, the reproduction is taking place. And next, economic importance is there that we have to study. Already time up. We will discuss the next class. Okay. Okay. Economic importance of the bacteria is there. So many diseases caused to the plant, diseases caused to the animal, diseases caused to the human beings. So many diseases which cause to the human beings: typhoid, cholera, dysentery, diarrhea, diphtheria, and then gonorrhea, syphilis. So many diseases which are causing by the bacteria to the human beings. Okay. And so many plant diseases, you know, the crown gall and citrus canker, you know, yellow vein of the bendy, and so many diseases which are causing by the bacteria, even paddy blast also causing by the bacteria, and angular leaf spot of cotton, angular leaf spot of cotton disease, cotton leaves, cotton fruits, angular leaf spot of cotton, and potato scab, potatoes, crown gall of potatoes. There's so many diseases are caused by the bacteria. These are the harmful effects. degradation of the food material also cause infection of the food, botulism like spoilage of the food material, and so many importance also is there. Bacteria, hormones production, enzymes production, vitamin production, even in the food production. In the food production, we use hormone production. We use in the enzymes, so many enzymes production, so many organic compounds, citric acid, vinegar, so many various productions also we use. So many enzymes productions, vitamins production. We use that in agriculture also as a decomposer, as a fertilizer. It gives it absorb the atmospheric nitrogen and it provides the nitrogen and phosphorus. The soil will become fertile by humidification, by decomposing the dead organic matter. Soil will be fertile. It will be bacteria will be phosphate in the carbon cycle, nitrogen cycle, phosphorus cycle, and also ammonifying bacteria, nitrogen bacteria. Already studied nitrosomonas and nitrobacter, nitrogen fixing bacteria. Azotobacter is aerobic and autotroph. That means it's not depending upon other. It is not symbiotic, non-symbiotic aerobic bacteria, azotobacter. So many exams they ask the question. Aerobic nitrogen fixing bacteria, azotobacter. The remaining are anaerobic, but azotobacter is aerobic and non-symbiotic. Non-symbiotic aerobic nitrogen fixing bacteria, azotobacter. Rhizobium bacteria is a symbiotic bacteria. We know rhizobium root nodules, and that it form. And some of the rhizobium, even the branchia also, azotobacter. In industry, in the so many production, cheese, bread, milk, ghee, curd production, even in the vinegar production, citric acid, vinegar production, vitamin production, especially B complex vitamin, K vitamin, so many vitamin production. In the retting and tanning industries, for the separation of the jute from the plant, retting industry, the tanning industry, separation of the leather from the dead bodies of the animals, for the separation of the leather from the dead bodies of the animal, that is tanning industry. Retting industry, fiber, extraction of the fiber. And so many alcohol production, organic compound production, even in the degradation of the petroleum and the ocean. Sometimes if the ships maybe leakage, all the water surface will be occupied by this. All the water surface will be occupied by oil. That oil will be separated, split by Pseudomonas phytidae. I already told you, debugging bacteria, very, very important. Even in the medicine, antibiotics productions, streptomycin, terramycin, conamycin, chloramphenicol, so many antibiotics production. The vaccines, in the vaccines preparation, we are using the bacteria, plasmids in the vaccine preparation. Even in the wastewater treatment, also we are using the bacteria, Pseudomonas bacteria. Insects, biological control of the insects. Bacteria, Pseudomonas, bacillus bacteria, which kill the insects, which kill the aphids, insects, as a biocontrol agent. And also, so many food spoilage caused by the bacteria and the water pollution, and also denitrifying bacteria, which again convert the nitrates and nitrites into again nitrogen. And so many diseases which are causing to the human beings: typhoid, cholera, tuberculosis, leprosy, pneumonia, diphtheria, rheumatism, and gangrene, dysentery, gonorrhea, syphilis. These are all the diseases caused by the bacteria. Leprosy, pneumonia, tuberculosis, kshaya, kushtu. These are all the diseases. Diphtheria. Diphtheria means, the tetanus, tetanus, diphtheria, the bending of the bones, infection, especially at the infection time, injuries time, if you not take antibiotics, that means injection, TT, penicillin injection, it cause for the tetanus, diphtheria, diphtheria, motions, dysentery, loose motions, gonorrhea and syphilis, sexual diseases, sexual transmitted diseases. So many anthrax disease to the animals. Goats, sheep, so many goats, sheep, and cows are died because of anthrax. Anthrax war also happened. Bacillus bacteria spread in our India to cause damage. Like that, biowar. The biowar also, even corona also one of the biowar. Coronavirus. Bacillus, olden days, bacillus is the biowar. Bacillus is spread in the air. So many animals, kettles will be died because of the anthrax disease. Plague, sometimes plague disease also happen in our India. That also. You see, so many plant diseases, black rot, and so many spot diseases, wilt diseases that are caused by the bacteria. You see, necrotic blight, crown gall disease, wilt diseases. These all are the leaf spot diseases, root, carrot rot, how the carrots are damaged, potato, how the potato are damaged by the bacteria. These all are the economic importance of the bacteria. Okay, in the next class we will discuss the viruses. Next class we will discuss the viruses. Okay, there is a useful and harmful aspects of the bacteria is there. Okay, in agriculture, industry, medicine. Okay, in the three aspects, there is a importance is there. Importance of the bacteria is there. Okay, agriculture, industry, medicine, and then diseases caused by, even in the environmental pollution also we using the bacteria, and the harmful aspects of the bacteria. This is also one of the essay question. Economic importance of the bacteria. Economic importance of the bacteria is very, very important. Okay, we'll stop with this. Okay, the next class we will discuss the virus. Okay, right.
Thank you, Shri.
Okay, bye.